Okay, hello, and thank you all of you for joining us on the Young Women in Bio YouTube channel. My name is Christina Herbert, and I am the chair of Young Women in Bio in Southern California, which is based in the San Diego and Los Angeles areas. This event was the first event of 3 experiential learning sessions hosted by Young Women in Bio Southern California, entitled Genomes, Germs, and Jewelry. And this is part 1, which is entitled DNA on Display, and was recorded on July 1st, 2020. And so participants in the live event were shipped supplies to follow along with a hands-on activity that we directed. So if you'd like to follow along with this activity, you're going to need some supplies. And so to extract your own DNA, you will need the following items. First, you need dish soap, pineapple juice, just a few drops of each, some table salt, again, just a couple pinches, and alcohol, generally isopropyl alcohol. There you might need probably not more than 5 mils or so. You'll need a plastic tube that holds about 5 to 15 mils of liquid and closes tightly. So in the lab, these are known as Falcon tubes. This is the tube I'm showing here with the little blue lid. But of course, any sort of tube should work. Generally, it should close tightly so you don't wind up leaking stuff all over the place while you're shaking things up. You'll need a dropper or plastic transfer pipette that might look something like what we're showing here. And then what we supplied students with was a black cord necklace and a small bottle charm vial. So it's a little vial that is a charm that can be hung on the necklace. And this is the little bottle that you're going to put your DNA into, and you're going to put that on the necklace, and then you'll have your own DNA necklace. So these items should be pretty readily available online anywhere that you look. So please, if you'd like to follow along, please get these items, and I hope that you will enjoy this event. Here is the infographic that we supplied our participants with. On this sheet, you can see the various steps that you'll need to do as you extract your DNA. And then finally, this is the back of the infographic that we provided to students, and this explains some of the science behind each step. So, of course, as many of the students following along found, the activity doesn't always work. And, you know, science is a lot like that. Sometimes it doesn't work. And some tips are to first ensure that you have enough saliva in the tube when you're starting. One of the tips was to maybe gently bite your cheek, something like that, just gently, to try and maybe slough off some extra cheek cells so you get a few more cells into your saliva and hopefully some more DNA. Another thing that was noticed is that the color of the dish soap used influences the color of the DNA extracted, or just slightly. But so if you want to extract something that looks a particular color of your choice, you know, go and find some dish soap that that has a color that you like. So, and of course, if it doesn't work the first time, try it again. This is the way science is. You can vary some of the different steps and figure out what works best, and just keep on trying and figure out what yields the best results for you. So with that, I invite you to watch the original DNA on Display event, and I hope you enjoy it. Thank you so much for joining us. I'm going to do a bit of an introduction before we get to the formal part of the presentation. So I want to tell you a little bit about YWIB. So our mission is Young Women in Bio gives girls today the inspiration and support they need to become tomorrow's leaders in science, technology, engineering, and math. And we strive to provide education and hands-on experiences in STEM, as well as share our passion for all scientific fields. And so YWIB Online is a new initiative that we've started as a result of COVID-19. And this is a new way for us to reach people and young women across the country and Canada. We have 13 chapters across the country and one in Montreal. So we really hope that by having these tutorials online and these events online, we can reach even more girls that are located in areas that are not close to any of our chapters. So we support women nationally, like I said, and we partner with a lot of leading companies and universities, hospitals, and other organizations to host these sorts of engaging and educational events and to try and motivate young women to pursue STEM. In 1 year, we had 70 events across North America, partnering with 80 different organizations and reaching, we had 304 presenters, but we reached over more than 4,000 girls. And we're hoping now that with our events being online, we can reach even more. So like I said, we have 13 chapters across the United States and Canada. So you can feel free to attend any of our virtual events now across the country. So I know most of you are from Southern California, but you can certainly check out some of the events from our other chapters. And then, nope, it's not going to advance. Okay, so but before we get started, I wanted to tell you all a little bit about some other initiatives that YWIB has. So unfortunately, this YWIB Ambassador Program, it was just launched in the spring, and unfortunately applications were due yesterday. So it's a bit late to get involved in this now, but I wanted to make you aware of it for next year. The YWIB Ambassador Program is for young girls to get involved and serve as an ambassador to their community and to their peers, and to support us in bringing events and programs to young women across the country. And so you would be partnering with our chapter to help us lead these sorts of events. So we're really excited to have this program started, and we are going to have our first, now that we have the applications in, we will have our first event, or our first ambassador, starting in the fall with us. But then besides this national initiative, the YWIB Ambassador Program, we have another program that Southern California is launching, and this is Southern California's Biotech Influencers Program. And this is going to be a monthly webinar series to discover your abilities and your future in STEM. And so our idea here is to have you engage with thought leaders, innovators, and industry experts who are going to aim to bridge the gender gap in STEM, and have you realize your full STEM potential. So we want you networking. We're going to have sessions where you'll get coaching and be able to learn more about what it is you're interested in STEM. You'll earn a shareable completion certificate if you participate in enough of the monthly webinars, and all will be provided, of course, free of charge, as all of our events are. So this is a program that's geared towards high school students. So you should be looking out for the registration process to begin in late August, and the program will then launch in October. Okay, so with that, I wanted to get started on the actual event and introduce our speakers. Like I said, this is Genomes, Germs, and Jewelry, and the topic, or the title for our first event in this series, is DNA on Display. So our presenters today are Biopolis, and Biopolis is a student outreach organization out of Canyon Crest Academy, and they are located in Carmel Valley, in San Diego here, and they are a group of high school students that got together to create science outreach. I'm sure you guys have given me a much better write-up of who you are and what you do, and you will introduce yourselves in a second anyway. So I'm sorry I don't have my notes, but they put together a lot of great programs for students in the San Diego area, so that they're going to be leading us through the hands-on activity today. So you'll get to see the types of activities that they bring to schools around the San Diego area. And this is a really great program that they've set up. A lot of their events are free of charge, and then they also have a summer camp that they put on where they charge, but they have a wonderful series of hands-on activities that they bring to young students. So I know we have some younger students on the call today. So if you're interested in those types of activities, you should check out Biopolis. And then we'll follow up the hands-on activity with a presentation from Kjerstie Bourne, who is a sales representative at Elim Biopharmaceuticals. And she's going to tell us a little bit about DNA science, what DNA is, what it can be used for, what sort of careers involve knowing about DNA. And she's going to tell us a little bit about her career in science as well. She has a unique path through her scientific career, and she'll tell you more about that. So with that, I think I just have one more slide here. I have to thank our sponsors. So New England Biolabs and Scientist.com helped us to pay for all of the supplies you're receiving and the shipping so that we were able to send these to you, and so we could bring these sorts of activities in a new way to our group. You know, we had bought all of these supplies with the hopes of having an in-person event, and that got canceled, of course, but now we are able to bring that to you virtually. So it's wonderful to see all of you. And with that, I want to start with the speakers. So thank you for joining us, everyone. Make sure you're staying safe out there. And once the video is available, we will edit it and put it on YWIB online for all of you to see. So if you, what I had wanted to say is if you didn't get your supplies, the supplies will eventually come with an instruction card. So you can follow just the instructions that are in the packet, or you will be able to see the video once it's online, and so you can follow along anyway. Okay, so with that, we're going to get started. Okay,
thank you so much, Christina, for that introduction. So hi everyone, my name is Caitlin.
I'm Julia.
And first off, we're going to start by sort of talking about what we do as Biopolis, just so you guys can get a better idea of what it means to be a science outreach organization as high school students. So I'm going to share my screen, and do you guys see Biopolis? Okay.
looks great.
Great, thank you. Juliet, take it away.
All right, so what is Biopolis? So Biopolis is a student-run organization that engages underserved youth in San Diego with exciting science experiments in order to ignite an early love of STEM. And the reason we started this was because we think it's really important for kids to be introduced to science in a way that makes it seem engaging, interesting, and fun, and a lot of students don't have these exposure opportunities. And we also are hoping to build diversity, both socioeconomic and racial, in science fields, because as it stands right now, San Diego and California and the country as a whole could get better at promoting diversity in STEM fields. Next slide. So we kicked off our existence as Biopolis with a fundraiser, which came in the form of a summer camp, in which we charged kids in our neighborhood to attend a biotech camp, and we charged them a nominal tuition fee, and we've been using that money ever since to fund programs for underserved youth in San Diego. And so we do that in a number of ways. So the main way we do that is by connecting with other nonprofits in San Diego. One of those is Solutions for Change, and that organization helps homeless families escape their situation and try to build a better life for themselves. So Biopolis goes to a lot of their events where the parents are either attending programming or classes, and we teach science to the kids, and we do just kind of short, fun experiments with them. And here are some pictures from some of those events.
And then another organization that we work with is Children's Initiative, and they essentially provide enrichment resources for underserved schools in San Diego. And we mainly work with Children's Initiative to launch after-school programs. And so I mainly wanted to talk about a specific one that we did in Vista, California, and it was targeted towards grades 4th through 6th. And so kind of the transition between elementary and middle school, we think that that time is the most impressionable for children. And if they develop an interest in science during that time, they're much more likely to pursue it in high school and have a much more successful career. And so we taught them fundamental biological concepts and procedures using hands-on experiments. And so just like a really brief overview of an example of our curriculum, we taught them microbiology, the structure, function, and location of DNA, the cohesive properties of water, cells, and biochemistry, bacterial transformation, genetic manipulation, and then DNA evidence and how that's used in forensic investigations, bacteria like E. coli, and then also gel electrophoresis, and that's an example of a procedure that we teach them. And this is some more pictures. This was the bacterial transformation where the bacteria was glowing. And this was our makeshift gel electrophoresis that we DIY'd with the kids. And so how we're adapting to COVID-19, right now we're working with Solutions for Change specifically to assemble experiment kits where we provide all of the materials necessary and send it over to where the families are living. And then we also pre-record a video with a lesson and also a worksheet, just so that the kids can continue to get quality science education, especially during this time. And then finally, we are also in a position where we have gained enough experience that we can teach staff to how to do what we're doing as well, specifically the staff that work with Children's Initiative. And then finally, like Christina mentioned, we are holding science classes this summer. And so for those of you who are around 5th through 8th grade, so we're targeting like basically middle school grades, it would be so wonderful if you guys could sign up, or anyone who has younger siblings or friends or relatives that have younger siblings, etc. You can go to this website at the bottom, and I will put that link in the chat soon. And basically what we're doing is we are adapting our curriculum to like sessions that we're going to be holding over a period of 4 weeks. And so we'll be teaching you guys chemistry, biology, and physics through hands-on experiments the way we have been doing. So yeah, hopefully you guys can look through that link and see if you guys are interested. And that's pretty much it for introducing Biopolis. So now I wanted to introduce each team of Biopolis here, specifically to show you guys our journeys with science as high school students, just so we can get a better idea of how like each person can explore science their own way. And so I will start. Hello again, my name is Caitlin, and I actually graduated high school, specifically Canyon Crest Academy, just like a couple weeks ago. And in the fall, I'll be attending UC Berkeley, majoring in molecular cell biology, and I'm also on the pre-med track. And so I actually did a lot of scientific research in high school through lab internships, and Julia and I will get into how to kind of get those a little bit later. But yeah, so what really hypes me up about science is working in the lab and like the hands-on aspect of it, but then also the process of discovery and that, you know, elated feeling when you discover something about, for example, like the cells that I was studying that no one else has discovered before. Yeah, so that's why I love science, and that's how I'm involved in science.
Hi, I'm Julia Picker. I also just graduated from Canyon Crest Academy. I'm going to be studying molecular and cellular biology at Dartmouth College in the fall. Like Caitlin, I also did a little bit of my own research in high school. I interned at a pharmaceutical company called Mirati Therapeutics in La Jolla. And I also interned at a San Diego State University lab. And this kind of piggybacking off of what Caitlin said, like my favorite part of science is just the idea that you could be pursuing something that's so exciting and so new, but also at the same time like doing something that's going to really benefit the world, and like discovering things like that, like vaccines, for example, because we're definitely in need of one of those right now, that can really have a profound impact on a wide amount of people. So that's really what attracts me to the field of science.
Hi, I'm Kaylee Mafong. I'll be a junior next year at Canyon Crest Academy. And throughout high school, I've done a little bit of genomics outside of school. And so that included some DNA extraction, which you guys will be learning about soon. And I've done a little bit of PCR through that as well. And one of my favorite things about science is just getting to work with other people and work together towards a common goal, which I think that discovering things for the future is just really important and a great skill to build as a kid. And that's why I love science.
Hi, my name is April, and I'm going to be a junior at Canyon Crest Academy next year. So I started really getting into bio just this year, actually. I'm taking a class for the USA BO, and I'm now part of Biopolis. And my goal ultimately is to become an ER doctor. And I think the thing that draws me in about science is that it explains so much, so many mysteries about life. So because I'm someone that really likes to understand things, and science can really help me like understand how life works and like why we're walking around, and just yeah.
Hi, I'm Lily. I'm a rising senior at Canyon Crest Academy, and I'm going to be leading Biopolis next year. And so as curious as I am, I enjoy exploring science experiments at school, and I was always fascinated by the results. So with Biopolis, I had the opportunity to share my fascination with science with others. So sometimes I study science not only in school, but also in my own time. I'd bring like a friend over and we'd do labs or fun experiments at home. And throughout this year, I've realized that communicating with others and sharing scientific concepts with curious and engaged students makes me really happy. For me, the best part of Biopolis is that it brings people together and it expands opportunities for the youth to explore STEM.
Okay, thank you. So now we will be going into the internships that we were talking about previously. And so just to go more in depth, I have interned at UCSD, the Salk Research Institute, as well as Stanford Burnham Previs Research Institute. And that was through my sophomore, junior, and senior years. And the main way that I received these internships was through, first of all, reading scientific papers. And so all of these labs, they write papers based on the research that they're doing and report their findings, their discoveries in science. And some of these discoveries, as I read them, get me super excited, or I have really interesting questions that I want to learn more about and find answers to, for example. And so I will find the lab that wrote these papers and then email them basically saying, I know I'm a high school student and I don't really have the skill set maybe that you're looking for, but I would really appreciate it if you could teach me. Because I think especially in academia, labs are really looking to also serve their community, give back to their community. And they can do this through teaching high school students who are essentially the next generation of scientists. And so offering yourself as a really interested and someone who's really engaged through your email can be really impactful and could actually land you an internship.
To piggyback off of what Caitlin was saying, I interned at a pharmaceutical company called Mirati Therapeutics in my junior year of high school. And then after my junior year, during the summertime, I interned at a San Diego State University lab. but I'd say that something that was useful for me was that the summer before my junior year of high school, I did a program called Cosmos through the UC system. If you guys haven't heard of that, I would highly recommend looking it up, because it's a really, really strong program for kids who are looking to go into STEM fields. And there's a whole bunch of different types of science that they teach there. I did synthetic biology, and I learned a lot of the fundamental skill sets needed to work in a lab. And so I was able to, when I sent emails to labs, I was able to say, like, I have a little bit of experience working in this lab. I studied under a couple professors at UCSD. And then another thing that I want to emphasize is the pure volume of emails that you send in order to try to get an internship, because I probably sent out, I think, 9 emails to professors before I got one to take me on in their lab. And so I would definitely say that if you're feeling discouraged by like maybe 1 or 2 rejections, do not give up. Just keep going, because at some point there will be a professor who wants to take you on. So I would definitely emphasize the large sample size and like getting emails out and sent. That would be my best bit of advice for getting internships in high school, if that's what you're looking for.
Okay, so there are also other ways to get involved in science aside from internships. Like at school, a lot of schools offer clubs and projects dedicated to different aspects of science. For example, around the same time I joined Biopolis, I joined a club called Aquascope, which mostly deals with environmental science and sustainability. And so the benefits of that is that you get to meet new people who have similar interests, and you guys can collaborate toward the same goal and even create like your own curriculum. Schools also feature science magazines that you could join, and if you're into like competition, Science Olympiad is another good one to get started with. And for me personally, volunteering with Biopolis has really helped me improve my communication skills. It's been a really enjoyable experience, and it really makes me want to continue to pursue science. And if you're also thinking about joining your own club, it's possible to expand your project by working with other organizations, such as Young Women in Bio, what we're doing right now, because it enables you to see science in a different way when you work with other different projects.
Okay, finally we can start our DNA extraction experiment. So can everyone get their materials together? And Julia will be leading the demonstration, so she'll be doing it live on camera. And then Kaylee, April, and Lily will be explaining the science behind each step of the procedure. And I will be screen sharing the sort of slides that explain the science, but hopefully Julia will be the one that's highlighted. Okay.
Okay, hi guys. So one of the materials that you guys are going to be using today is dish soap. So dish soap cleans dishes by removing fats, and the same process occurs during DNA extraction, believe it or not. So DNA isolation begins with lysis, or the breakdown of tissues and cells, and this allows the fats called lipids and proteins that make up the cell membrane to be dissolved. In this case, the dish soap separates the fat and the water, and this allows for the DNA to be released from the cell as nucleic acids from the nucleus. Next slide. So pineapple juice is added in order to further dissolve the cell membrane, and it contains enzymes which break down any remaining fats on the exterior of the cell that dish soap doesn't break down. The juice further degrades lipids, which allows for better isolation of the DNA.
Okay, so next up, after the pineapple juice, we have, next slide. You have the salt. So after that membrane has been broken down, the salt separates the DNA from the rest of the solution. So it clumps the DNA fragments together so that it neutralizes DNA's negative charge, making it less soluble in water. And then after the salt, you mix the solution, right? And so you mix the contents together to allow the ingredients to come into contact more frequently and act on the DNA. So as you mix more, you increase the frequency of collisions between the ingredients, and that increases contact.
So next you'll be using some alcohol, and the main purpose of alcohol in this experiment is to precipitate the DNA. So this process is achieved in two ways. First, DNA has an overall negative charge, so it precipitates by reacting with the positive ions in solution. And since alcohol has a lower dielectric constant than water, meaning that it's unable to keep positive and negative ions apart very well, it allows the DNA to go through with that reaction. And second, since the alcohol can hydrogen bond with water molecules, it leaves less water available to hydrate the DNA. Which allows it to reach the positive ions in solution and form a precipitate. Next, please. And last step is to extract the DNA. So due to intervention from the alcohol in the previous step, DNA has formed a solid precipitate and becomes visible to the naked eye. As a result, because DNA is insoluble in alcohol, the fragment folds in on themselves and clump together, separating from the solution so that they can be taken out from the tube.
Okay, so now I'm going to quickly guide you through the experiment. So you should have all of your materials with you. You're going to need a tube to put your spit in. I've already spit in this so that you didn't have to watch me do that on camera. You're gonna need a few drops of dish soap, pineapple juice, salt, alcohol, and your necklace that you should have been sent along with your materials packet. So I'll give you just maybe like 30 seconds to get everything together, and I will get my stuff together. All right, I'm gonna go ahead and get started. So the first thing you're gonna do is you're gonna spit your saliva into the test tube. The best way to do that is just put it right there and spit it in. It's gonna sound gross, but we're gonna put soap in it right away, so it's gonna be completely a-okay. You don't need to worry about coronavirus or any of that stuff with this. So after you do that, you're going to take your dish soap. I have it in this little thing right here, this little tube. And we're going to take the dish soap pipette and put in 2 to 3 drops of dish soap into your spit container. I'm just gonna put that right in. I'll give you guys a second to do that. You can flick it to mix it just a little bit. The next thing we're gonna be adding is the pineapple juice. So you should have been sent some pineapple juice as well. You're gonna take your pineapple juice pipette, and that could just be any clean pipette that you're gonna allocate to pineapple juice. And you're gonna take, oh, no pineapple juice, okay. Never mind, we're gonna skip the pineapple juice step. The next thing we're gonna do then is we're gonna add a few pinches of salt. Now I'm just gonna eyeball this and just dump a little bit of salt in there. Then you can sort of flick it to get it mixed up in there. And then you're gonna take the cap of the tube. You're just gonna kind of invert this up and down a couple times. And this just gets everything this gets all the reagents in contact with the DNA in your cells. So after you've done that a couple times, yes, Caitlin is saying you can use contact lens solution in place of the pineapple juice if you have that. But if you don't have either of those things, the experiment still should work, or you could use meat tenderizer. It just might be a little bit more difficult to see the DNA. So once you've done that, you're going to take a new pipette for the alcohol. You're going to need quite a bit of alcohol. So you're going to just grab a whole bunch of that out of there. Isopropyl alcohol, which is, I know, in short supply these days because of the virus. And you're just going to add about 15 to 17 drops down the side of the tube. And like they said before, that's going to help us separate the DNA out of the solution so that we can actually see it. Very important step. So now you're probably not going to be able to see anything just yet, quite yet. So you're going to flick it a little bit more until you see a sort of white stringy clump start to form. In mine, I don't know if you guys are going to be able to see that, but it just went to the bottom. But there's like a little clump floating around in there. That's my DNA that we extracted from my cheek cells. So the next thing you're going to do, you got to be very careful with this because you don't want to get all this disgusting stuff everywhere. You're going to take the necklace that you were sent, and if you can see right here, there's a cap and then a vial. So you're just going to pull the vial off of the cap, and the cap's going to stay connected to the necklace. And you're going to take a clean pipette and transfer that little bit of DNA that's in here. Oh, how much pineapple juice? 2 to 5 drops of pineapple juice. And like they said, that's just going to help us see the DNA better because it further breaks down the cell membranes and the enzymes that it contains. So if you want to do that, you can do that at any point in the procedure. It's okay if you haven't done that yet. But I'm going to continue with our little task of transferring the DNA. So you're just going to make sure that you get as much of it as you can in the pipette. Then you're going to slowly add it to this little vial right here. You might not be able to see it very well because I'm covering it. But what do we put into the vial besides the DNA? Water. So there's going to be, when you transfer, there's going to be a little bit of that extra like water, salt, alcohol mixture, but you'll be able to see just a little bit of DNA in the bottom there, and it'll be more evident when I put the cap back onto the vial. We're going to sort of just put that on and give it a few flicks to shake it up. And there should be, if you did it right, there should be some clumps of DNA in the vial. Let's see if you guys can see that, I hope. And there it is. You've made a necklace out of your own DNA. And do not worry, this DNA cannot be used to clone you or anything like that. DNA is pretty finicky in that it denatures. And we've probably done quite a bit to destroy some of the structure of it already by adding all of these ingredients, but we can still see it when it clumps up together. Yes, it is awesome, Emily. Thank you. Now you can just make it into a necklace. Oh, you guys actually have it connected on both ends. And so in order to actually make the necklace out of it, you have to take this little, you should have received it. Here, let me show you guys. You should have received it connected and all on one end of it. So what you're going to do is you're going to thread this end, the one that doesn't have any metal on it. I've already put this together, so it's already done, but it shouldn't have any metal on it. You're going to thread it through this little loop thing, the one that's free, and you're going to take either a hammer or some pliers, and you're just going to smush it on there. You're going to smush it around that little string. And don't worry if you don't get it the first time. It's kind of difficult, but you can just take a hammer to it, or you can get some scissors, or you can get some pliers and just just get that on like that. And you can have a parent help you with that too. It's not super easy, but I trust you guys will be able to figure it out. And so, yeah, you've got your DNA, or a tweezer. You've got your DNA in a necklace. And that's the end of our little demonstration. I hope you guys enjoyed.
Just to and I also wanted to show you the tenacity, I guess, of DNA. So I have my own right here. And yeah, but this is my DNA from a year ago. I think it's pretty cool that it's lasted so long. And that's because the alcohol really maintains the integrity of the DNA.
Hi, everybody. This is always fun. I really enjoy talking about science, and I'm so glad to be here with you today. A little background story about me. When I was in high school, probably around most of your ages, I got sick with valley fever. And I was really sick from the disease. And at the time, there wasn't much known about it. And my dad was a professor of sociology and ran an applied research center. And he felt really bad watching me be super sick. And he decided to devote his time and talents to organize and get together a bunch of other experts with himself to find out more about the disease of valley fever. He linked in with MDs and PhDs, and together they conducted a comprehensive study. I thankfully recovered. The study did leave me with one thing, and that was a love of science. And even though it was sad to be sick, it was a great experience to be able to learn from some amazing scientists in the process. It's really awesome that we can use science to help people, and really that's one constant that I find with most scientists. They're all really kind people who want to use their knowledge to help others. DNA is super fun, and I'm old, so the story I told you did not include DNA sequencing. But if we were doing the same study today, we would have definitely been using DNA sequencing to find out more about the disease that I had. I like to set expectations during the call presentation, and today we're going to be going over what is a CRO, who uses DNA, what DNA looks like, and what is done with the DNA when a company like mine, Elim Bio, gets it. The goal is after the call that you'll be hopefully able to appreciate DNA and DNA science and appreciate how science can help other humans on our great planet. As Christina mentioned, I've had a vast number of jobs within science. Science has been a really great career for me. I've been able to change what I'm doing based on my needs and my family's and children's needs. It's been interesting, engaging, and also a flexible career for me. Currently, I work for a CRO. A CRO is a contract research organization. A CRO supports science on a contract basis. This means that my company does not do our own research. We research for other institutions. A CRO reduces costs and speeds up work for scientists. My company has robotics technology that is expensive to purchase. We run day and night so that when researchers need the results, they can get them faster. A lot of times they will submit a sample to us at the end of their workday, and then the results are ready for them when they get there the next morning. Lots of research facilities use a CRO. It's a great symbiotic relationship. I really do love what I do. My days are filled with helping other awesome researchers. I get to make science a little easier for a lot of great scientists out there. And I get to talk to them. Usually when it's not COVID-19, I get to visit them in their labs. I get to be really creative with my communication. And it's really fun to get to know some amazing researchers and hear about what they're doing and help them a little bit in the process. So who is using DNA? DNA is used in the medical field. An example of that is my aunt had breast cancer. And thankfully this was over 5 years ago and she's doing great now. She found out she had breast cancer, and when she did, they checked to see if she had BRCA1 or BRCA2 mutation. These are genes that are inherited. So people who inherit these genes tend to develop breast cancer and ovarian cancer at younger ages than people who do not have these mutations. She was so happy to tell my cousin, her daughter, that she did not have BRCA1 or BRCA2 mutation. Since it's inherited, It would be possible if my aunt had these genes, my cousin would also have them. DNA is used for genetic testing to find out gene mutations. It's also used for medical diagnostic to test, diagnose some medical conditions. Another example of the use of DNA is DNA fingerprinting. Forensic scientists use DNA fingerprinting to identify criminals and find out if the evidence that's left at the crime scene links with DNA from the criminal or potential criminal. DNA can be taken from blood, hair, fingerprint samples. Police investigators and forensic experts are able to use the data in matching the DNA found at the crime scene with the DNA of the suspects. DNA profiling is used successfully to positively identify mass casualty incidents, bodies or body parts in serious accidents, individuals and victims in mass war graves by matching it to family members. If you're wondering, I'm not this cool, I don't know a lot about this topic, but I do have a college friend who works in the FBI, and I can tell you if you're interested in this type of work and you're happy to be and lucky to be a female, then you're more likely to be accepted into the FBI as a female scientist than any other path. Okay, and another great thing that people are using DNA for is family tracing. I'm really lucky that I know where my grandparents came from. I've been able to track their ship manifest. They came in from Norway on Ellis Island. I could track which churches they were at in Norway, and I can even go back to my family farm that's been in my family for over 200 years. But most people don't have these kind of details, and this is where family tracing really comes into play. I know a couple people who've had really amazing experiences with family tracing. One of my friends is adopted, and she wanted to actually know her medical history because she didn't have that much information about it. So she did one of these tests to discover her medical history, and at the same time found out she had a sibling she never even knew she had. And now they talk and they get together once a year, and that's really awesome side effect. Another person I know was actually contacted by a daughter she didn't know she had. They were even actually on American Idol sharing this story. The woman had donated eggs, and later, thanks to family tracing, found out she had a daughter. And they had a really fun parallel life. They were both singers, and even actually attended the same university and were in the same choral group many years apart. Lots of people like to use DNA family tracing to find out more about their background and their history, and just kind of find out more what their past was like. DNA is also used for anthropology, paleontology, and archaeology. DNA mutates or changes over time, and these changes can be compared. DNA changes in organisms, and the changes are passed down, which allows us to figure out historical evolution. Side note, when you've been in the science field for a really long time, you get to know some really cool people, and most scientists are really great and like to share their work. And I was lucky to get to visit and stay at an archaeology dig for a weekend. And I can tell you that the digs are awesome and very dirty. Biotech researchers also use DNA, and this is mostly who is sending DNA to my company. Researchers are usually placed into two camps, industry and academic. I know people that work in both areas, and they're all really awesome humans. Both industry and academic research have amazing breakthroughs and help our society improve. It's also so fun to work with somebody that has made a great discovery or created a new drug. It's just really fun and exciting. The goals of academic and industry are at the core the same: to discover and help other humans. The funding, focus, and what drives their goals is what's different. Industry goals are set by market needs. A great example of this is right now we need a COVID-19 vaccine, and the industry has lots of companies working on this. Industry sometimes has competition like that, where multiple companies are working and racing towards the same goal. The structure within industry is that there's a great amount of teamwork. Lots of researchers are on the same team, and everyone's working towards the same larger goal. Research and academia goals are fueled by procurement of grants and for the continuation of research. Academic researchers usually wear many hats, and in addition to researching, they also publish papers, teach courses, mentor students. Industry and academia are both a vital arm to scientific innovation. All right, now a little bit about DNA. DNA, make sure you remember this awesome structure, is short for deoxyribonucleic acid, and it's famous for its double helix shape. Inside the double helix are the instructions and essentials for all forms of life. DNA has 4 nucleotides, cytosine, guanine, adenine, and thymine, or A, T, C, G. And A and T always pair together, and C and G always pair together. Some people like to compare DNA as like a recipe book, but I prefer the analogy of computer coding. I'm sure you know how a computer is coded on binary, with on being 1 and off being 0. And everything we can do on the computer is done using these codes. So just 2 things, on and off. And we can do music and art and writing and videos like this one. And the list really goes on. And all of these things that we can create on the computer can be saved, copied, and shared. And DNA is amazing because instead of just having 2 ways to write the instructions, a 0 and a 1, it has 2 types of base pairs, AT and CG. And the cell reads the DNA on 1 strand of the DNA in 1 direction. Therefore, each base pair represents 4 different values that the cell can read through the DNA, and twice as much as a computer bit. So it's pretty amazing that the DNA is found in every cell, and all this information is available to be read. What does the DNA look like when we get it? I like this happy DNA. That's how I kind of imagine DNA coming to my company, happy and helpful. When my company gets the DNA, it comes in many different forms. One wonderful thing is, one way we get it is bacterial colonies on agar plates. And these I'm hoping maybe you saw in science class already. And another way that we get DNA is from bacteria pellets. And the pellet is made by spinning the DNA really, really fast. And the liquid rises to the top, and then the pellet remains. Glycerol stalks can be also stored for a really long time, and liquid bacterial cultures are added and to the glycerol solution, and it makes them structurally stable. And plasmid DNA is put into a circle, excuse me, and those are commonly used for research. And then this is a 96 well plate, and these are a very common way for us to get DNA. There's lots of samples all labeled together. And usually what happens when we get these plates is they're labeled with letters on the top and then numbers on the other side. And then that kind of helps us know what each sample is so we can refer to it like C5. And they're very tiny little wells, and they hold lots of information and bacteria. All right, when we get the DNA, what do we do with it? And what we do with it really at our company depends on what the scientists ask for. Because like I mentioned, we are just instituting and taking in what the scientist wants and processing the information for them. A common thing people ask for is PCR, and I'll go over that some more in the next slide. PCR is a way that we can make copies. Mini prep is a way to identify the target DNA within the host bacteria. A mini prep limits the contaminating proteins in genomic DNA. RCA stands for rolling circle amplification. This is where copies are made kind of like PCR, but it's for plasmids. And then sequencing, I love sequencing. It's super fun. It's really amazing that we can figure out each nucleotide and bases and figure out what order they come in. And this is kind of what it looks like when we process sequencing order. And I can't wait for you guys to learn about sequencing next week. I believe that next week you're going to have someone here to speak about sequencing. And then DNA purification is another thing. If a contaminated DNA sample is used, there's a higher probability that we'll get erroneous results later. So a lot of times we do DNA purification. All right, PCR. I told you I'd tell you a little bit about this. PCR uses reagents or things that we add. One reagent that's added is a primer, which are short strands of DNA, also called oligos. And these primers have complementary sequence to the DNA region. And we make these little oligos for people so that it's exactly complementary to the DNA. Another reagent that is added is DNA polymerase. DNA polymerase is an enzyme that's needed to replicate the DNA. Almost all PCR applications employ a heat-stable DNA polymerase or Taq polymerase. In the first step of PCR, the two strands of DNA, double helix, are separated at a very high temperature, and this is called denaturation. And then in the second step, annealing, the temperature is lowered and the primers bind to the DNA, and then the two DNA strands kind of become templates for the DNA polymerase to assemble a new DNA strand. And then for the next step, elongation or extension, in this step, DNA polymerase can add the free nucleotides to the DNA strand. You'll see that this strand is adding in one direction and the other strand is adding in the opposite direction. This is because DNA strands are anti-parallel, which means they are parallel with each other, but they go in opposite directions. And PCR methods rely on thermal cycling, and this is repeated many times, 20 to 40 usually. And it can take a couple hours to complete. So you can see why researchers might want to give this job to somebody else. The target DNA can go from just one or a few copies to billions of copies and has exponential growth. An example of this growth is starting with one DNA strand, and I have my one DNA strand over here as an example. And then we run one PCR round and we end up with 2 copies. And then we run another PCR round and then we have 4 copies of our DNA. And then we run another PCR round and then we have 16 copies. You can see how it's exponentially growing. And then another round of PCR and we have 32 copies. And this process, as I say, can take around 2 hours to complete. And the PCR is checked for quality and quantity. And we do that using gel electrophoresis. DNA is negatively charged and the DNA is pulled through a gel matrix by an electric current. And this separates the DNA fragments according to size. So here is, oops, sorry about that. Let me go back here. Here's a video about DNA replicating. Oh, good, you can see it. So the DNA, as you can see, it's going up to 95 degrees Celsius. That's about 203 degrees Fahrenheit. And then it splits apart. And then we're gonna drop the temperature back down to about 72 degrees Celsius and then 55 degrees Celsius. Typically, during thermal cycling, we denature at 95 degrees for about 30 seconds, and we cool down and anneal for about 45 seconds, and then we extend for about 45 seconds. And as you can see, it keeps expanding, and then we're going to heat it back up again and repeat the process again. Down, add our primers, add the Taq polymerase, heat it back up again, separate them up, and this process goes over and over and over again. All right, so next I wanted to share a little bit about gel electrophoresis. Let me just fix my slide a little bit for you. So gel electrophoresis is a very common process that is done. As I mentioned, the PCR is checked for quantity and quality, and the results of the PCR reaction are usually made visible by using gel electrophoresis. DNA is negatively charged, and the DNA is pulled through the gel matrix by an electric current, and this separates the DNA fragments according to size. And this is a photo. As you can see, there's a DNA ladder, and that kind of helps us know how far and what size the DNA is that's traveling. And as the current is applied, the DNA moves through. And if the DNA is heavier or has more base pairs, it moves slower. And if DNA is lighter or has less base pairs, it moves faster, it moves farther along the ladder. I wanted to thank you so much for having me here. It was really fun to talk to you guys and share what I know about science. And I wanted to share, if you want to know more about PCR, there's a really great video on Khan Academy about it that I do recommend. And stay curious, wash your hands, friends.
Thank you so much, Kjerstie, for such a wonderful talk, and Biopolis also. Those were really excellent. Both of you gave terrific presentations. And so with that, this ends the formal part of our presentation. And I'd like to say a big thank you both to Kjerstie and to the young women from Biopolis for their amazing talks and leading us through the DNA extraction protocol. Another big thank you to our local Southern California sponsors, YWIB sponsors, and then our national sponsors also. And finally, I'd like to thank you for joining us, and please stay safe, everyone. We're all in this together, and look for more virtual events and important information on our Young Women in Bio online webpage. With that, we are going to say goodbye to the people who are joining us on YouTube, and those of you watching live should join us afterwards for a Q&A session. Thanks so much. Goodbye, everyone.